Okay, so we're ready to start. And hello, everyone. And so welcome to our electrochemical colloquium. Today, we're discussing electrochemical processes in biochemical systems and how they can be studied computationally, and then how to relate them to non-biochemical systems. So some of you may be noticing that there is an increasing number of groups these days who put their research efforts into integrating electrochemistry with biochemistry, with enzymology and cell biology. And there are many reasons for this. But the only thing that is very clear is that this interface is clearly developing with a lot of interesting fundamental questions to be answered. So our today's speaker is Professor Arieh Warshel, who pioneered the field of multi-scale modeling for complex chemical reactions, and who's a leading expert in computational enzymology and biochemistry. So Arieh received his PhD in 1969 at the Weizmann Institute of Science in Israel. He also did his postdoc at Harvard University. And then in 1972, he came back to Weizmann Institute of Science and worked together with the Laboratory of Molecular Biology in Cambridge in England. And then in 1976, he became a faculty at the Department of Chemistry at the University of South California. Arieh, thank you so much for joining us today. It's a really big honor for all of us to be hosting you here. Now the stage is yours.
Okay, thank you very much. I'm happy after all the work that I put last night to kind of summarize some of the work that we've done in what could be called really electrochemistry in biology, where in many cases we have to invent the methods, since I did not trust too much continuum approaches, though in general they are correct. So the idea was always to have a microscopic view, but one that could work with the computers of that time. And as you will see, a lot of the approach could be used to both chemical and biochemical systems. So I will go and move between different type of systems, and probably most emphasis will be on ion channels, but I will consider other systems in some details. So when we go to this electrochemistry, one of the key questions is the storage. I mean, whatever we do is dealing with charges. And what I forgot to say is that in biochemistry and biology, almost everything is controlled by electrostatics. Some people view different systems as batteries and capacitors and so on. So you have to be able to develop the approach to study such systems, and obviously they are relevant to non-biochemical systems. So charge storage, the issue is evaluating redox potential. And during the year, it started really in 1974, I developed different methods for calculating the energetics of charges. And it always moves with the computation of the given time, but we are still using some of the methods that were developed in 75. So there is hierarchy of approaches. You have the fully microscopic approach where you have the charge, its surrounding, and you basically go up to infinity with the proper boundary conditions. This is used with free energy perturbation, which we start to use in 86. And in 75, we introduced the Langevin dipole model, where the water was represented explicitly as dipoles on a grid, and they were trained to reproduce the correct solvation energies. This model was highly criticized by people who had no problems in using continuum with unknown dielectric. And then there is a PDLDS, where we project this Langevin dipole model to a model where the protein has a dielectric. This is called PDLDS. This model is basically the only model that provide a clear definition of what is the protein dielectric. It's not the dielectrics that you get by actually evaluating the macroscopic dielectric, which we also put a lot of effort. Okay, so with these models, we started in the 80s to study redox potential, where the first system was cytochrome C, and we tried to figure out the redox, and one of our main trick is always to calculate the difference between being in water to move into the protein. In this way, we don't have to figure out the redox in water, though we also develop approaches to do this by QMMM approaches. But in general, the question was how the protein changed the redox. The slide disappeared yesterday, but we were able to reproduce the redox of cytochrome C quite nicely. And in a later stage, we moved to the problem of iron-sulfur proteins, where there are many proteins with very different redox and sometimes with almost the same environment. So using this Langevin dipole model, we were able to calculate the redox basically almost from first principle, of course, relative to the redox in water. And we got quite early a very impressive agreement. Here we see it just for some iron-sulfur protein where there is a very large difference in redox, and it's all due to the different environment and its ability to stabilize or destabilize the charge. So here, for example, we have cases of kind of the same cluster in almost the same active site with very different change in redox. And it's really due to penetration of one extra water molecule near the cluster. So we use this redox calculation in many, many problems. One of the nicest use was in studying the storage of light energy. We could call it photochemistry, photoenergetics, and where the main direction was photosynthesis. So it's the issue of converting light energy to charge separation energy. The aim at the time, which was started essentially at the early 80s, was the bacterial reaction system center, trying to understand how light excites this system and how it converts the light energy to charge energy. So there is this famous dimer, and the charge separation could go either, in principle, either to the left or the right. And what we try to understand is what happened in the first 3 picoseconds, where you end up with plus on the dimer in the upper place and minus on the pheophytin. For this, we develop a whole series of approaches. In each case, they were developing the first time. A free energy perturbation to calculate the energetics of different states, because the charge could be either, in the first step, go from plus on the dimer and minus on this chlorophyll monomer, or going directly with superexchange to the pheophytin. Most people favor at that time the pheophytin, and we say we cannot figure out from experiment what happened, and we must use reliable calculations. In addition, we develop a surface hopping approach in 81, which allow us to calculate the time dependence of the electron transfer, and also to evaluate the Marcus parabolas from first principle. In this approach, you run dynamics on the surrounding, let's say the protein, and you calculate surface hopping approach to see what is the chance to move to the next state. And 2 weeks after we got the structure, which was a major issue, we have all the machinery ready already, and we figure out that the first intermediate is actually dimer and chlorophyll rather than superexchange, but it is hard to see it because it exists for very short time, and then you go to the P plus H minus. Another direction, which is really of interest for general electrochemistry, was the idea of this time to put different donor acceptor through the membrane and to get nice photoelectric device. We study it kind of theoretically and essentially conclude that if you have the same donor and acceptors, this will never work, because you will have the dielectric barrier, because it doesn't matter what you do. There is no magic of instant exchange between these chromophores. You will have to actually climb the energy of charge separation and have a barrier that amount to putting the charge in low dielectric. We basically argue that if you try to do this process in water, to have a chain of donor and acceptor in water, the problem will be large reorganization energy and it would not work. If you try to put identical donor and acceptor in the membrane, you will have this dielectric barrier, and therefore you have to use donor and acceptors with increasing electron affinity, which is what is done in photosynthesis. Some people argue that this is trivial because we know what is done in photosynthesis, but at that time nobody had any clue what will happen if you actually put such chain in a membrane. And we suggested experiments of putting chain of acids through a membrane with varying pKa and creating interesting photoelectric device. So this is kind of a part which usually people don't read too much on the energetics of this charge separation. Now, what could have been a whole lecture is considering electrocatalysis, because when you have a catalyst, when you have a electrochemical device, you need to apply catalysis. We spend huge effort in studying enzyme catalysis. And in general, our approach involves studying the chemical reaction in solution. This is, for example, GTP and magnesium surrounded by classical water. This is a QMM ab initio simulation. And we study the surface for this complex system. And once we understand the chemistry in water, we take the ab initio surface and convert it to empirical valence bond surface, which is much, much faster. Use the empirical valence bond surface in water, so we reproduce the ab initio, and then we move the system from water to protein. This is a case of a GTP as protein, where we try to study the hydrolysis of the GTP. And here you have a water that transfer a proton to the phosphate with the help of magnesium. And then we have bond breakage in a chemical reaction. What is much more important is that we could use free energy perturbation umbrella sampling to calculate the energetics of this reaction in the native and mutant protein. We have many, many studies and reviews on this problem, but we could study what is the effect of mutations, sometimes mutations that cause cancer, and also decipher different mechanisms. We use this approach to understand the origin of enzyme catalysis. Again, very large amount of studies, and we show, but what you see here is the barrier without the enzyme, which sometimes is enormous, and the barrier after the enzyme work. And we show that in all the cases that we touch, the catalysis relative to water is due to electrostatic effects. So obviously, you could use charge stabilizing environment both in biochemical and chemical catalyst. Now, another issue which is related to electrochemistry is molecular motors. It applies to motors which are built by chemists, and of course, our main aim was to understand biological motors. So a system which we studied very extensively was the ATP synthetase, which has a head where the chemistry of ATP to ADP is the fuel that allowed this motor to rotate, and a lower part, F0, where the transfer of protons from one side of the membrane to another allow this ring to rotate, and it kind of lead to synthesis of ATP. So we want to understand this system on the same level that physicists understand this system. I mean, this is a famous exercise in first year physics, like how much work is being done by pouring water and lifting this weight. The result disappears, so you could do the calculation yourself. And in order to study it, we develop multi-scale coarse grain model where we simplified the protein, replacing the side chains by spheres. This idea was introduced by us first in 1975, but in later years, we taught this system to do electrostatic, and we trained the model on folding energy and other features, got the right energetics for membrane proteins. We validated it on a lot of systems, and we use it on many systems, but one of them was this ATPRs. And in ATPRs, the question was reproducing the result of experiments where people like Kinoshita bound a bead to the head, to the stalk of the ATPRs, and measure the rotation in real time. And these great experiments gave strange results, because instead of rotating 120 degrees in each stroke, it moved 80 degrees and then continue the rest, where the chemistry occur between the two steps. It's very hard to figure out how this works experimentally, but when we have these coarse grain free energy surfaces, it's much clearer. And what you see here is on one axis, the different states of the motor. So this is the chemistry. And on the other axis, the rotational position of the central stalk. And what we see here is that you could move 80 degrees without any barrier. Then if you want to move to the next step, you have to do the chemistry, and this is done in the region of 80 degrees. This is kind of a special catalytic dwell, this is how it's called. And we reproduce this, we reproduce the energetics of the whole system. And we also provide for the first time understanding of how the energy of the chemistry is converted to rotation. At the same time, our competition, when they try to study this question, apply force on the stalk, which could give you any results that you want, instead of having a model where the chemical forces creating a surface which then move the stalk. So we study other related systems like myosine, and in all cases, what you have to look for is the trend in electrostatic energy. This is the overwhelming part that drives the motor. Now we go to a system which kind of resembles the most a battery. And this is the system of ion channels. And first, a very short introduction. I mean, we want to figure out how some protein or another entity in a membrane responds to the application of potential on the electrodes. And when I studied this system, which is very complicated, I, of course, read the continuum papers, which led to great contributions, but I never trusted in any stage, including the development of enzyme models, the continuum picture, because I never felt that this is really what I could comprehend on a molecular level. So always strive to something where I could see the representative of molecules and ask how the system works. And of course, insist that it will reproduce experiments. So ion channels, as you know, are everywhere in the body and in the cell. A very large part of medicines are aimed at different ion channels, like in heart disease, many other systems, and of course the ion channels activate or control the brain and control how much you are listening to me now. Now, we start to study ion channels quite early in 1990, where the only thing that exists was gramocidin, and we waited very impatiently to the emergence of a biological ion channel. When the potassium channel emerged from the study of McKinnon, at some stage we entered trying to simulate the current. At that time, it was already 1 year because it took us time. Other people tried to use atomistic models, which did not include the barriers and miss many other features. So first we use, in the first few years, the Langevin dipole model and evaluated the free energy profile along the channel. Now, what we really want, and what I always demand to get, is the observable, which is the current, and also the difference between potassium and sodium. We done it, where is the model? By having the potassium channel embedded in a model membrane, which involve grid of polarizable atoms, and Langevin dynamics that drove ions through the channel. We run simulations. This model, since we use Langevin dynamics, allow us to go to long time. We run simulations of potassium current and of sodium current. Typically, like the potassium simulation look like this. The green is the chlorine. The color one are the potassium. They are in different color, so we could follow them. And eventually, the red one, which is in the cavity, will be kind of pushed out by the next ion, as you see it now. Not yet. And then it will go out. Now, when we study this, and we reproduce essentially the conductivity of both the potassium and the sodium, we found that in the case of potassium, we have a 2-ion events, whereas in the case of sodium, each ion pass alone. Now, we translated this to a concept which is still not widely appreciated, that the difference between potassium and sodium in terms of conductivity is due to different charge dielectric. Now, I wrote very extensively on dielectric, tried to change misconception of different people, but this will not be discussed in this talk. But charge dielectric is really what phenomenological dielectric you will put for the interaction between the ions if you want to describe the microscopic reality. The more the environment rearrange, the larger will be the dielectric and the closer the ions could come. So what we concluded is that for sodium-sodium, the dielectric is smaller, the charge dielectric, than for potassium. And I think this still holds as a fact. Now, the next stage was to move to more details and to study voltage-activated ion channels. This is closer to the battery model. And again, you could do it by very strong computers or specialized computer like the Anton computer, or as we do frequently, by more simplified but approaches that could give us the insight in a faster way and in some respect a better way. So we choose, and in some stage we will move, and we are moving to more computer time, but even now we still use an approach which are based on simplified things like using Langevin dynamics instead of atomistic dynamics. Now, what we try to study is these ion channels, which when it's closed with the polarization of plus up, minus down, the ion does not pass. But when you change the cell polarization, it opens and the ion pass. We want to study it by microscopic rather than macroscopic model, because as I already mentioned, I don't like macroscopic models, and I will mention some of the shortcuts. And this slide, by the way, belonged to the previous story on the Langevin dipoles, but it got stuck here. So when we want to study the system with Langevin dynamics, we need the friction. And of course, there are many ways to get the friction by simulations. There are frictions which include memory function and a lot of stuff. We don't like any of them because, for example, when you are stuck, when you have a very complicated system, when you're stuck in different sites, it's not clear what friction you will use and what you leave to the potential of mean force. So we introduce what we call renormalization approach, which we validated very extensively, where we run dynamics under the influence of strong force, so the transfer would happen, on the microscopic system, and then run Langevin dynamics with different frictions on the simplified model. And we change the friction and change the forces until both approaches gave us the same results. I still think it's by far the best approach to get a friction for your model. Some people could think differently. Here it's, for example, used for opening and closing of proteins, where you apply very strong force on a real protein, apply the same force and a friction on the coarse grain model and get the best friction. Okay, now with this model, we also need what might be more important, the model of the system. Now, as I said, I did not like the continuum model, despite their enormous contributions to ion channels. In each case, they involve assumption, and some of you might know even that the continuum model don't even have the electrodes, and they have major problems in formulating the gating charges. So we decide to have a model where everything is explicit. Of course, we have a membrane, protein, and the main element was to have the electrolyte represented by grid of points whose charges determined by the field from every part of the system, the channel, the electrodes, and the other grid points. This model, which took us a long time to build, was validated. This is typical system. It has different regions because when you're at infinity, you don't want to have grid points. So it was validated by reproducing kind of all known test cases. Later, I will show you that it was also validated by explicit model. And with this model, we reproduce the gating charge, which is the effective charge that is being formed when you just start the channel before the current goes through it. In this coarse grain model, you evaluate the charge by taking all the grid points and summarizing their charges in both sides of the membrane. These are different systems. You do it for different voltage. These are details of the model, which if you read the long papers, you could figure out. And one of the nicest work with Il-Soo Kim was to take the potassium channel to try to get the landscape of closing and opening, and then to calculate the gating charge in each of these states. But obviously what we are measuring is the gating current. So how do you get the current from the charge? Very simply, you know the time that the system moves between the different states. Oh, what is this?
You can just click OK. It should be fine.
I'm afraid because this ate yesterday my presentation, but okay. So since we know the time of moving between the different states by knowing the barriers, the current is the change of charge divided by the change of time. And we got this gating charge as function of time. We reproduced this nice experiment of Benzelia, and we got very significant insight. Of course, you always want to validate on a more advanced level. Just a minute, we'll check the time. And we decided at some stage, I think it's 1916, to try to get validation by having more microscopic model. And we move to Monte Carlo with the primitive model. Primitive model is when you have ions that interact by Coulomb law, and of course, van der Waals to prevent collision. And this is in JFIS Chem, either 1916 or 1918. And we were able to reproduce more or less the same results of the coarse grain model. We did it even for the potassium channel, and this was quite satisfactory. At another stage, we did, I would say, one of the deepest work, that it's so deep that I don't fully remember it. This was done with Il-Soo Kim, and we tried to have kind of capacitor model which gave us huge new insight on what used to be the equation of the free energy, the relationship between current to voltage. And in the macroscopic model, you basically have to know the free energy of the process, let's say the opening. So it's not I would say, fundamental model. Here we try to use our kind of Marcus parabolas and to have on one axis the QV product, and on the other axis, the calculated free energy of the system. And also, in this model, the intersection is the barrier for the current motion. So it's a very, very involved. There are two papers, one in BBA and one is JFIS Chem. And it also kind of reproduced or gave very nice insight on the potassium channel. And it's highly recommended to read as a more physical view on the on the product of QV and on the fundamental of Hodgkins-Huxley model. Another interesting system that we studied, which again was major challenge for us because we did not know how to do it, was the question, how do you simulate the effect of applying voltage on a biological system? For example, there were very nice study of Peter Brzezinski and also George Fair on the application of external voltage on the bacterial reaction center. And this approach supposedly could give you the direction of charge migration, which is very important, for example, in a system like cytochrome C oxidase, where you don't know really what is the path of the charges. So what we knew to do is the generation of voltage when you separate charges. But the opposite process was not clear to us. And essentially, I put this puzzle on a wall of one of my smart colleagues, and I also put $5, and I say that if he tell us what to do, he will get the $5. Now he is Dean, and I did not get the $5. But this is the system, bacterial reaction center. This is the experimental setup. And what we came up at the end was the following idea. Since we don't really know what to do, we will apply different voltage, calculate the charges, and then do it for different voltage. And we will take the voltage that give us the lowest free energy. So in this way, you reproduce the voltage generated by a given charge separation. And it works very well. And we intend to do it on cytochrome C, or we kind of did preliminary studies. And what we found is that you cannot use this voltage as it is because it gives very similar results for different pathways, which intuitively you would not guess. So these are different applications of this capacitor model to biological system, and of course it could be applied to batteries. And the last example is proton transport, which, for example, if you want to have hydrogen battery, this is what you want to study. You also want to study how the proton goes through different electrodes and different separators. So we were working, or I was working on proton transport since 1978. Model it mainly by the empirical valence bond, which unfortunately I have no slide here, but you have parabolas for reactant, parabola for intermediate, parabola for products. You could calculate the curvature of the parabolas by evaluating the reorganization energy. You mix the parabola to get the actual adiabatic free energy. And since the model is so simple, you could use it for very, very long simulation. However, while it's very effective for enzyme, if you have proton transport across a whole cytochrome C oxidase or across bacterial reaction center, it's not so useful. So we came up with another simplification. We have several water molecule or donor and acceptors described by explicit EVB, but the rest of the system is introduced classically, which anyhow happen in the EVB. And then you run dynamics on it, but you run Langevin dynamics, and you watch the motion of the proton. Of course, we did our validations. The friction was determined from the autocorrelation of the energy gap. One of the main systems for validation was carbonic anhydride, where you have a proton transfer in some mutant from the water bound to the zinc farther away to histidine or lysine. And in this stochastic Langevin dynamics, we could follow the time dependent of the motion of the proton, and we got a very nice reproduction of the observed results. We did major validation on grammacidin, where we reproduced the proton current at different pHs. Again, I'm not aware of anybody else who did such studies. We also debunk the Grotus mechanism that a lot of people were proposing as the most important feature in proton transfer in biology. We have no problem with it in water, but in biology, the proton transfer is controlled by the energetics of the intermediate states. Again, this is a subject of another lectures, but we show it on aquaporin, on grammacidin, and many other systems. Now, in some cases, like cytochrome C oxidase, this Langevin dynamics approach is not effective enough. So we came up with time-dependent Monte Carlo. I just show here that for each problem, we have to find the right approach. And in this time-dependent Monte Carlo, we know the time for basic proton transfer between donor and acceptor in fluctuating environment where there is no barrier. When there is a barrier, we add it as Boltzmann factor, and then we run Monte Carlo that gives such nice, this is moving in the D channel in cytochrome C oxidase. I already lost the proton, but I think it was here or it is here. Okay, some of you saw the proton. So we could study very long distance proton transport by this approach, and we are still using it. The last system, which is kind of interesting, and again, a system that is of great interest, is this HV1. It's a voltage-activated proton transport system, which has very significant medical interest, a lot of beautiful experiments. What you have here is arginine in an acid, and you try to transfer a proton. Now, there were different theoretical studies of this system, and here kind of come my insight to such systems. I cannot accept study of new complicated system where you do not reproduce the whole observed process. I mean, you could reproduce it incorrectly, but you don't try to do it. The published studies, some of them did quantum mechanics of proton transport. One tried to look on the voltage. None of them had any clue of how you reproduce the actual proton conductance as a function of the applied voltage. So we done it by combining EVB profile with our channel model. And we found a very exquisite application where you have this arginine and the aspartic. In the closed form, the arginine is far away from the aspartic, and the proton is stopped by the arginine. But when you open the channel, okay, when the channel is closed, the proton plus cannot pass, it repelled by the arginine. But when you change the polarization, now the arginine moves away, and now the proton could pass. I think it's up. Okay, now it passes. It goes through the aspartic and go to the other side. Now, so here you have to combine different theoretical approaches, pKa calculations, EVB in the case of the moving between aspartic to water molecule, and the external field, which we also validate by calculating gating charge. And we show, it's a relatively complex paper, but very beautiful result. We show how the barrier is really erased in the open state. And we also show that unless you model the chemistry, you cannot really get the correct picture. This is a kind of a complicated picture which explains the coupling between the external field in the proton transport. Here are some of the people, co-workers who were involved in different studies, which I thank all of them. And this is a book that describes these studies and describe my trajectories. And again, thank to all of you for listening.
Thank you so much, Ariel. It was a very interesting talk. I think it's really interesting to see how this complexity of the problems entail the complex or requires the complexity of the approaches, and that in each case you have to come up with a different solution.
Yes.
We're open to questions, so the audience can raise a hand or you can unmute yourself and ask a question. And maybe in the meantime, if somebody wants to ask a question, go ahead. But in the meantime, maybe I will ask a few questions that are a bit general, but just curious about your opinion. So in general, in enzymes and in molecular motors, what is your opinion about the role of the solvent, the environment, the solvation sheath that is formed around? So is it a very big contribution energetically, typically, or very small one? Because that determines how you simulate that part.
2 weeks ago, I gave in Berkeley, there was Molecular Frontier Symposium on water, where this was the subject. So in general, if somebody interested, it's recorded somewhere. Molecular Frontier is a kind of organized by Nobel Foundation, and it's for young people to get insight into science. Now, the rule of the solvent is as follows. If you do not include it in the calculation, as was done by many prominent scientists when they try to study photosynthesis, you get complete nonsense. So it's a part of the system which must be included. Now, once you could, however, include it on many different levels. It's not essential to include water molecule as a real water molecule. You could include it as dipoles, you could include it in CG model. So it is a part of the system, it has to be included. In some cases, the solvent, like in some enzymes, you have few water molecules that play crucial stabilizing role. In other cases, they even participate in the reaction. So your solvent must be included. One could say that the water molecule in proton transport are part of the solvent, and we have to figure out how to place them. We have for these water flooding approaches, but so the answer, the solvent is very important, in particularly when you don't include it.
I see, but would it be correct for me to say that the biggest role that it plays is when the molecule or an ion desolvates when it enters the enzyme or enters a channel? That's when you have a barrier that is the biggest, and that comes from the solvent part.
You are right, but you could say that if you try to calculate binding energy and you don't calculate the energy of the ion in the solvent, you get error, which is 70 kilocalorie in the case of aspartic acid.
Mm-hmm.
So this is, I mean, you could rate the role. You see, people in the early day, before many of you were born, but it was quite recent, in my opinion, talk mainly on the role of the solvent in structural effects, fluctuations, and so on. And I have a huge difficulty publishing eventually in 79 this dipolar surface constrained dipolar model, because people did not realize, I mean, people who look on temperature effect of solvent did not realize that the energetics of the solvent is much more important.
Mm-hmm.
So it's, yes, it's what you and me say. The solvent is very important, but not the detail, but the detail of the solvents are really not so important.
Okay, interesting. So then the role, because you also discussed the role of environment in the redox properties, and that you show that there is a big role of environment, like where exactly the redox at which potential takes place. So it means that in some cases the environment, the solvent, can play a huge role, and in some cases it can be on the secondary to the actual enzyme, on the actual biomolecule.
Yes, but what happened, one of the reasons why people, you would not believe it, but even in Kirkwood-Tanford model, which was a monumental work, they forgot to look at the self-energy, and even they wrote, Kirkwood, that charges are not created nor destroyed in biological systems. So I forgot what I want to say. I want to say that there is huge compensations that when you move from the solvent, the new environment always try to give you almost the same solvation. And this is why the role of the solvent was ignored by some.
Okay, interesting. Because I mean, the reason why I'm asking some of these questions is because in electrochemistry, so when we work with heterogeneous electrocatalysis, I can move my microphone closer. So when we work with heterogeneous electrocatalysis, there the simulations, there is a lot of debates about which approach to use, explicit, implicit. Last time we had a long discussion about this also with a different speaker. And so looks like, so the molecular approach to explicit is of course the best because it takes into account a lot of things. Implicit
but it depends,
works, but only in some cases.
It depends on the computer. It always depends on the computer power.
Yes.
Yes.
Yes. So this is why I'm curious about how you, because what you showed in the presentation, you showed different approaches. So Monte Carlo, coarse-grained, explicit approaches
yes.
too. And in each case, you had to pick a different one. So is there any sort of general logic that you can say, you can deduce from all this work about which way to go in terms of the solvent simulation?
Yes. Okay, so it's really a time-dependent answer.
Okay.
That in 1975, some people study solvation by having one sodium and one HCl. And this had huge impact, where people did not understand that this has nothing to do with solvation. I mean, people just miss the issues that you have to get the totality at the given time. Now, when you have faster and faster computers, of course you get, it's better to use a explicit all-atom model, but there is still a problem. Let's say you simulate ion channel and ion current. And again, the more we wait, the more explicit approach are progressing. But if you run just one simulation and one free energy calculation, you have this issue of convergence that you don't, even if you run long run, you have to check again and again to see if it works. So unless you could, if you run 2 months simulation and you get a result, you will have the issue of trying to understand where the result came from.
Mm-hmm.
And in simpler model, it's easier. I always believe in using both of them. I mean, you use the simplified model, validate it, use it several times a day to try to get the insight, and then you also run the full microscopic. So it's an issue. I mean, I think that you get a lot of understanding from fast run. Now, if you could run every day a run of, let's say, GPCR or something, fine. So again, the longer we will wait, the less relevant will be my approaches to try to get it on small computer in a short time. But but I'm always very happy to argue with people who want to use explicit because, and again, you also have to know to calibrate.
Yes, yes. But for the implicit ones, you also have to do a lot of parameterization. Right?
No, of course.
No, no.
I meant to calibrate the implicit.
The implicit, yes, yes.
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 And also to calibrate the explicit is less concerned. The explicit, you need, it's the issue of the time.
Mm-hmm. Okay, maybe the last question that I would like to ask you is related not to the solvent, but to the redox center. In your opinion, what is the role of the rigidity of the structure? And so the dynamics, how much do you have to really, how much of a role sort of it plays in the charge transfer processes? I know that there are very rigid enzymes and there are those which are rather flexible sort of structurally.
When you say rigid, I mean, I view it only as reorganization energy.
Right.
And which play major role in like electron transfer, you try to reduce it. So this is one redox, which is feature which is built in electron transfer protein. Part of it is is done in a very trivial way, like in cytochrome C, you just put it in a non-polar environment. This is not a great accomplishment. But in others, when you want to stabilize the redox center, you have to have environment which is more reorganized less than in solvent. So this is the rigidity that I think has a role.
Mm-hmm. Mm-hmm. Yes. Okay, I think that if we don't have any more questions, maybe we will wrap it up for today. Thank you so much. Ariel, was very, very interesting talk, and I think I hope it will stimulate the electrochemical community as well, because again, there is an overlap and it's pretty clear, but I think the overlap currently is happening more at a sort of a device level, but going into the bioelectrochemistry and connecting it to the real electrodes and maybe measuring real quantities, this is something that might be happening later, I guess. Maybe not.
So thank everybody and have a rest of the nice day or evening.
Thank you so much. Thank you.
Bye.
Bye.